Okay. Thank you for joining today. Ask Me Anything. We see this format as an important channel for direct engagement with our shareholders and the community following TuHURA Biosciences. Before I get started, I'd like to remind you that we will be making forward-looking statements, and as such, we refer you to our SEC filings for more information about the company and its programs. Over the past several years, TuHURA has progressed from early platform research into a clinical stage oncology company focused on therapies designed to help the immune system recognize and attack cancers that typically evade immune recognition. Today's AMA is an opportunity to expand on the company's publicly disclosed clinical progress, discuss how these immune modulating strategies aim to improve responses to existing immunotherapies, and provide additional clarity around the clinical milestones investors should be watching as the programs advance. So let me tell you a little bit about how the phase 3 trial is progressing. This is the trial with IFX in first-line Merkel cell carcinoma, comparing IFX with Keytruda versus Keytruda and placebo. And the primary endpoint of that study is to show that we can improve the overall response rate. We've recently announced that we are increasing the number of sites from 25 to approximately 35 to help enhance enrollment in the trial so that we can meet our projected target enrollment completion date of mid-year next year. And with regards to the phase 3 program, there are several milestones that investors can expect to see over the next 12 to 18 months. I would say probably two of the most important, obviously, are completion of enrollment, which, as I mentioned, is targeted for mid-year next year. And then approximately 6 to 7 months after the last patient is enrolled in the study, we should be in position to unblind the study and determine if the trial was successful in a statistical significance improvement in the overall response rate of IFX Keytruda versus Keytruda alone. And obviously, if that study is successful, that is a key major impetus and value creator for the company, and it should position us to submit to the FDA those data along with the other data required for a biologics license application, so-called BLA, which is comparable to a new drug application or an NDA for non-biologics, i.e. small molecule pharmaceuticals. And those are key drivers in any biotech company. The success of a phase 3 study essentially says that your technology works. It is better than Keytruda alone. So that combination then can become a new standard of care in the frontline treatment of Merkel cell carcinoma. I think the ultimate success, despite meeting the primary endpoint of the study, is having that data support a license application and having the drug go through the FDA and receive marketing approval. And that ultimately is the success for any clinical regulatory program, but more importantly, it's success for patients who have Merkel cell carcinoma who currently only achieve about a 50% response rate with the standard of care, which is Keytruda or pembrolizumab. Importantly, improving that response rates offer patients something that they currently don't have available to them, increasing to 75% likelihood of having a response versus the current standard of 50%. And that really fulfills the mission of the company, bringing to market novel therapies for patients with unmet medical needs. We've had pretty good interactions with the FDA. I think that is witnessed by the fact that we worked with them on a very novel trial design, actually a first in my 33-year tenure in this industry. And as you may know, we worked with the Oncology Center of Excellence. In that study design, they took our data, which was published from patients who had progressed on frontline, i.e. in the second line setting, and moved it into frontline, given that it was encouraging enough to utilize what they call their Project Frontrunner initiative. So instead of being a second line therapy, we're now being tested as a first line therapy. And in addition, this trial is unique because when one seeks accelerated approval, i.e. the overall response rate endpoint, Typically, that requirement is then to have a confirmatory study that would usually use overall survival as the clinically beneficial endpoint. We're not required to do that. The uniqueness of this trial that we worked with the FDA was to have an accelerated approval endpoint for accelerated approval, i.e. response rate, and not having potentially a requirement for a confirmatory trial, because the key secondary endpoint in the study is progression-free survival. So when the trial matures, if we achieve the progression-free survival endpoint, that would satisfy the requirement for a confirmatory trial and could essentially take accelerated approval and allow the product to now have regular or so-called full approval in its marketing of IFX in combination with KEYTRUDA. I think there are a number of other applications, as you'll see, for IFX 2.0. As you may know, we have reported that we started a study where IFX is given to Merkel cell patients with Merkel cell who present without skin or nodal lesions. These are called Merkel cell carcinoma of unknown primary. So they don't know where it came from, but they have these large lesions in their maybe liver, lung, or in their abdomen. And when they're biopsied, they're determined to be Merkel cell. They're not eligible for our phase 3 study because we use a small needle for dermal, subdermal, below the skin, or nodes where they're accessible. So this study is looking at the feasibility of giving IFX into deep-seated lesions like in the lung or the liver or in the abdomen, using radiologic techniques to guide that needle to inject the tumor. Then they're giving KEYTRUDA just like they are in the frontline study that we're doing in the phase 3 trial. And this study is looking to show that it is feasible to do that is safe to do that, and that the response rate should be what we would hope to see in our phase 3 trial, i.e. 70% or greater. We expect to have some of those patients hopefully presented this fall at the European Society for Medical Oncology, the so-called ESMO meetings. And I think that is a potential driver for the company, because it really will provide some transparency and insights to potentially how the phase 3 study may be working. So we recently reported in a press release that we have filed an IND with the FDA to begin a phase 1b/2 study for TBS-2025, and that's a novel VISTA-inhibiting antibody. And as we have previously reported, VISTA is the only checkpoint that's expressed on leukemic cells, and is the primary mechanism by which leukemic cells evade immune recognition. So if your immune system doesn't see it, that's responsible for why the response rates are relatively low, but more importantly, the very short durations and patients relapse. And so we worked with the FDA. The FDA provided us very specific guidance on what they wanted to see. We had a meeting scheduled for next month. The FDA determined that a meeting wasn't necessary. In fact, they'll just give us written responses to the questions that we have provided them. We should have those answers sometime next month. And then initiate this study in early fall, like in the third quarter, approximately the third quarter of this year. This is an open label study. We're going to be giving TBS 2025 at different dose levels to patients who have relapsed refractory acute myeloid leukemia, so-called AML. And we're looking at the specific molecular defined subset, the so-called NPM1 mutated AML. And the rationale for that is these patients currently are treated with a drug called a menin inhibitor. About 80% of them don't respond. And for the 20% that do respond, the response is very short-lived, less than 5 months. And so that's an unmet medical need population. And as I mentioned, that mutation drives VISTA on the leukemic blast itself. And it's why these patients don't respond or have very short duration of response. So the phase 1 study may give us some insights to whether blocking VISTA on leukemic cells would allow the patient to have an immune response and in fact see so-called complete responses in patients who really have no alternative therapies. We should have that data, at least insights into that data, certainly early in the first quarter of next year. Obviously, I think we mentioned this in the press release. If we show that we can salvage those patients, meaning have them have a meaningful rate of complete response, we would take that data to the FDA and determine how many more patients at a biologically effective dose, as it's called, the so-called recommended phase 2 dose, would they need to see for that potential application as an accelerated approval pathway. When we are completed with that phase 1B component, even if we are pursuing accelerated approval as monotherapy, meaning TBS-2025 by itself, we would also look at putting it in combination with a menin inhibitor to see if we can improve its response rate and its duration of response. Ultimately, this market is very attractive, still has a large unmet medical need, and quite frankly, it's an area that I grew up in terms of blood-related malignancies, having trained and run a transplant program at the Fred Hutch in Seattle. So I'm excited about the prospects for this VISTA-inhibiting antibody. We are the only company that is pursuing inhibiting VISTA on leukemic cells as a development direction. We tend to, and this is kind of an interesting question, how do we evaluate additional cancer types or treatments? We follow the science, right? The translational science. If a tumor exploits a normal mechanism by mutating it to its benefit to drive the disease, i.e. the leukemia in this setting, I mean, that's a perfect example. NPM1 has a normal purpose in biology in terms of how it regulates division in our cells. When a tumor mutates that mutation prevents those cells from stopping to proliferate, i.e. they're leukemic. So that cause and effect is biologically correlated. And the question is, if you can inhibit it, can you in fact have a benefit, i.e. decrease the malignant aspect of that mutation? That's what we're hoping to show in AML. We also know in other indications in leukemia where VISTA plays the same type of translational role, for example, in pre-leukemia, or even in other forms of mutated mutations in leukemia, so-called FLT3 ITD. Collectively, that is 70% of all leukemic varieties. Yeah, IFX 2.0 definitely doesn't compete with checkpoint inhibitors. In fact, its total purpose is to make them work in the 70%, 80% of patients where they don't work, right? The underlying biology for why checkpoint inhibitors don't work is the same, whether it's lung cancer, breast cancer, or Merkel cell carcinoma. Tumors don't recognize, patient's immune system, doesn't recognize the tumor as being foreign. If you don't have an activated immune response, you don't have a checkpoint to inhibit. So you need to have activated T cells that recognize the tumor as being foreign. That's what IFX 2.0 does. It makes the tumor look foreign, specifically like a bacterial protein. That activates your innate immune response against the tumor. And anything that's foreign in that tumor is presented to your T cells, so-called adaptive response. Those T cells are now educated to what your tumor looks like. They can be activated by the checkpoints, right? That's why you give a checkpoint inhibitor. So it takes the brake off of those T cells, which essentially gives a very aggressive anti-tumor response. That's how checkpoint inhibitors are beneficial to patients. So this is complementary, and it doesn't matter if it's Merck's Keytruda, if it's Bristol Myers Opdivo, or Basencio from the other companies like AstraZeneca, et cetera. It will enhance their ability to work in tumors in which it currently doesn't work. At least that is the theoretical, the so-called hypothesis, both mechanistically, and we're hoping to show that in Merkel. If we show that in Merkel, that should be translatable to other tumor types as well. Yeah, this is always the challenge for biotech, not to let your spend get ahead of the progress and the milestones that create value for company and for its shareholders. Specifically, we have a very lean operational footprint. The industry has changed in the last 25 years. I've grown up where you had to do everything yourself, and your spend was, really disproportionate to the timing and the milestones, your so-called time value money equation, which is different for a small biotech than it is for a company like Merck. Merck can wait 5 years to get a phase 1 trial result. TuHURA can't, small companies can't. And so we leverage having executives that have not just experience in drug development, but experience in fiscal experience in capital management and deployment, in terms of fiscal responsibility of the vendors that they manage, with regards to timelines, milestones, and cash flow requirements. And I think when you put all of that together, and we talk about this time value money, it's very helpful for a small company to keep its overhead very low. In fact, our G&A is 12%, 15% of our overall spend. What does that mean to shareholders? It means 85 cents on every dollar is going directly to the development program itself. That's the true value creation, right? Paying salaries and paying for overhead doesn't really help if you're paying more than what you are contributing to the development of the product itself. And believe me, some companies do that. There are plenty out there that are spending 30, 40 million a quarter to do programs like we are doing for 3.5, 4 million a quarter. Yeah, so we have kind of multiple potential milestones over the next 12 months. The one thing that good biotech companies do, or biotech companies that have been through the ups and downs of both macro and economics, geopolitical climate, and capital markets, is to really have a risk diversified and managed portfolio. Not a single technology, not a single product, but multiple products and technologies strategically that fit in terms of the company's overall vision in this setting of overcoming primary or acquired resistance to cancer immunotherapies. And so we would have, for example, IFX 2.0 has its milestones over the next 12 months. We talked about maybe having data on the IFX study in deep-seated lesions in the fall. And then, of course, having completion of enrollment, our target to do that in mid-year, early third quarter next year, actually June, July, of next year. So that's just the IFX program. You can layer on top of that the AML program in TBS-2025. We're getting a safe to proceed announcement from the FDA to move our phase 1b/2 program forward, seeing some of that phase 1b data, seeing if there's an accelerated approval potential route based on that data within the next 12 months, and initiating the phase 2b component in combination with a menin inhibitor. All of those are key value drivers for the TBS-2025 program. And then earlier in the development pipeline are these novel antibody-drug conjugates, or ADCs, which is a total paradigm shift from the industry. We're not targeting tumors. We're not looking to essentially bring cancer-killing products to a tumor. We're looking at targeting through a very novel mechanism, the ability to shut off cells to prevent immunotherapy from working, i.e. removing that immunosuppression that is so prevalent, whether it be in leukemia in the bone marrow or whether it be in your lung tissue with lung cancer. And so that technology, its first application in leukemia, will have milestones this year showing that potentially that the conjugate of a delta opioid receptor antagonist linked to a VISTA-inhibiting antibody, i.e. the TBS-2025, may have a totally different beneficial profile in a humanized model of AML in a mouse. That would be a key milestone for us. If that is a successful venture in terms of its outcome, that could be submitted to a very prestigious meeting in the American Society of Hematology in December. Recall last year, ASH essentially recognized the novelty of finding the delta receptor on MDSCs and its ability to control that cell's capability of producing immune suppressing agents. That was given oral presentation, which is very prestigious position to be in at the society meeting. So that data may be available for the December meeting for ASH. If it is successful, we would then put it on what we call a lead optimized compound, would go into IND enabling studies in 2027 and first in human potentially in 28. So even that program has some key successful drivers in terms of value creation for the program. So now you can get the sense of the multiplicity of different products with different associated milestones. Yeah, so we've been very fortunate. In my prior companies, we have worked with so-called funds that do investments based on royalty streams, so-called monetizing the potential for the royalty of a product when it's approved. And so I approached our largest shareholder, and this is when our share price was devastated by short sellers. I mean, it was just a typical small company gets caught in a so-called short selling activity. And we would never want to do a highly dilutive financing when the value of the company should be, in my view, 250, 350 million at that time. And so our largest shareholder agreed to provide us a credit facility. So it's $50 million at 12% interest. It's interest only for 5 years. We don't have to take the 50 million. We can take it down as we need it. And what that allows us to do is manage our balance sheet and our cash flows in the most capital efficient manner, right? Our cost of capital today is very high if we went out and sold equity. If we think the stock should be $4 or $5, why would you sell equity at $2 when I can take a 12% loan and cover my expenses for the next 2 years? And so it allows the company to potentially build the value that is intrinsic to its technologies and the milestones that we hope to achieve. And when the timing is right, i.e., if you have the type of currency that is reflective of the value of the company, if you wanted to go out and diversify your shareholder base by bringing in some key fundamental institutions, you could raise capital at that point, but it would be much less dilutive than doing it at $2. If you did a raise at $5 a share versus $2 a share, you can do the math. That's much more efficient in terms of managing both dilution and managing your cash flow requirements to maintain the programs that we currently talked about and keeping them on track. Operational flexibility. I think I essentially referred to this previously. The operational flexibility is having the right team that knows how to, they're not really crisis, but when you have programs that are coming off its original forecasted, either whether it be enrollment or whether it be timelines for manufacturing, et cetera, understanding how to have a backup plan or a rescue plan, whatever you want to term it, so that if you do have a slip in timeline, it's not substantial, it's not materially impacting the company. And again, that really has to do with hiring the right people and allowing those right people to do what they know what to do. They're the subject matter experts. Even though at the end of the day, I take full responsibility, but I rely on them to do what they've been doing for the last 25 years successfully. And I think that provides the team to reshift strategy for the short term, whether it has to be shift strategy for the long term. If there's another product opportunity that fits perfectly in the portfolio, we are certainly open to additional acquisitions, bringing in additional value, creating candidates into the pipeline. That's been my entire history, having built a portfolio where 4 of the 5 drugs were successfully approved, and the last one essentially allowing the company to be acquired for just under $2 billion. I'm not saying that's the scenario here, but I'm telling you what my But my perspective is on portfolio development, strategically growth, and where the company valuation should be at the time when you meet some of those milestones successfully. I mentioned having a lean operational footprint. I would, challenge you to find a company with a phase 3 program progressing towards completion of enrollment next year, a phase 1b/2 program with a second product coming into a therapeutic area like AML, and an early stage development program with a novel set of ADCs with 22 employees. That's correct. I mean, our overhead is $250,000 a year for 10,000 square feet of space, half of that being wet laboratory space. About a third of our employees are in the lab. And so we manage that infrastructure very closely. We hire people when so-called buy versus make makes the most sense. So if consultants are costing us more than an employee would cost us, then obviously we look for the right person to take on those operational activities. But we do that at a time where it makes sense to do that. And so we will continue to manage the infrastructure as we need it. Obviously, you don't need a full-time statistician if they're going to provide some input on a protocol, and then you have 8 months before you need the statistician again. That's just a very simple example of how you manage infrastructure. Okay, in closing, obviously, we want to thank all of our shareholders for their support, their belief in the programs. We're passionate about what we do, and I'd like to thank you again for really thoughtful questions and spending the time to listen to our responses to them.